Welcome back. This should be our last section for Chapter 18. On the last one, I believe we left off talking about question number 39, which deals with cytoplasmic determinants and inductive signals. Both are things that affect growing and developing organisms early on, as early as fertilization with the cytoplasmic determinants, but with the inductive signals shortly after the embryo has started to go through mitosis a few rounds. So once again, cytoplasmic determinants are things that happen to be present inside of the original cell that ultimately dictate development. On the next slide, we're going to take a look at what I mean by this. And we see that inside the egg right here, we have a nucleus, and with the sperm we have a nucleus, two haploid cells. They have half the original number of chromosomes within each of the nuclei. And we see cytoplasmic determinants. We see these circles and we see these triangles, and these are arranged in the cell. It kind of looks like an even split down the middle of the cell. That's not always the case, though. These just happen to be a random distribution. And sometimes they're dispersed evenly, sometimes they're not, and this does ultimately affect a growing cell. These cytoplasmic determinants only really have a role if fertilization occurs, which would be the sperm fertilizing the egg, forming now our first cell in the zygote. This is just one cell. This is how all life, at least from sexual reproduction, begins. So now these cytoplasmic determinants, they begin to have a role as the cell goes through mitosis, as they do aid very strongly in development. So with our first mitotic cell division, we can see in this particular sample that this cell has a lot of the green triangles, but not so many of the little orange circles or yellow circles, whereas this one is the exact opposite. These will ultimately affect how this cell grows and matures and develops, which will be differently from this one. So these are cytoplasmic determinants. The second part of question 39 was asking about inductive signals and how those affect growth and development. And that's what we see on the screen here. We see that our zygote is now many cells, not many cells, but much more than previous. It's saying 32 exactly. That's pretty specific. Anyhow, inductive signals are things that are present in one cell but are dispersed to neighboring cells, and they affect the neighboring cell's growth and development, and induction. So we have an inducer right here, which is from this bottom cell in this image, and it is being sent across the membrane, and it's received by the neighboring cell, and those go to the nucleus and they affect how this cell will grow and divide. So that's inductive signals in a nutshell. They are signals from one cell that are transmitted to another cell, and they affect how that other cell grows. I guess these are kind of similar to hormones in that regard. And then we have determination. Determination refer to the events that lead to an observable differentiation of a cell. These final states of cells occurs when an organism is still an embryo, so still very small. Once the final state of a cell has been determined, which is determination, the cell will function specific to that form, and so will all cells produced from it later on as a result of mitosis. So what this means is if a cell is determined to be a liver cell, it's going to function as a liver cell from this point on determination, and so will all cells that are produced from it as a result of mitosis. So in other words, the fate of that cell will be determined in this process, determination. And the last section that we need to cover in Chapter 18 is a short section, but an important one nonetheless. It's dealing with cancer or irregular cell cycle control, which is what we see going on in our title right here. The gene regulation systems that go wrong during cancer are the very same systems involved in embryonic development. So we're going to take a look at that here. The types of genes associated with cancer. Cancer can be caused by a few things. First is a mutation to the gene that control growth and cell division, or mitosis. In the cell cycle, we have different things called checkpoints, and these kind of make sure that a cell is operating normally. That's the function of checkpoints. Well, cancerous cells or precancerous cells, they blow right past those checkpoints and they continue to do their own thing, which causes a massive growth called a tumor. And tumors they can go at different rates. Some go fast, some go slow, some are stagnant and can just be fine. So there's really there's a lot of guessing going on with cancer, specifically the tumors. So we can cut those out if they're kind of stagnant, or if we catch them at the early stages. I guess that depends on the type. But anyhow, what can cause these mutations to genes? Things known as carcinogens. We talked about the chemical factors earlier on, smoking in specific, but also environmental things like radiation from the sun that can cause UV cancer, or skin cancer, I guess I should say. Once again, if we catch the abnormal growth early on, in most forms of skin cancer, at least we can cut those out safely before they get too deep and get to the bloodstream where they can be spread throughout the body. So looking at cancer at the genomic level, we're looking at oncogenes and proto-oncogenes, which are tied to questions 43 and 44 in your handout. As we can see on the screen, oncogenes are cancer-causing genes, and proto-oncogenes are normal genes used in the cell's life. Keep in mind, when we're talking about genes, we're talking about sections of DNA right now. Proto-oncogenes are sections of DNA that are normal, and oncogenes are sections of DNA that are altered. So oncogenes, proto-oncogenes, they are polar opposites of one another. Oncogenes are formed from proto-oncogenes. Typically, oncogenes form as a result of mutations within the cell. This conversion is a result of one of three mechanisms. They are, number one, movement of DNA within the genome. So DNA can be translocated from one section of DNA to the next. Second, overproduction of a protein or overexpression of a gene. So if we use a gene to make too much of its product, that's overproduction. And the third are point mutations, which could cause a change in the codon sequence. Those three mechanisms I just mentioned are your answer for 44. Number one, movement of DNA within the genome. Number two, overproduction of a protein or overexpression of a gene. And the third, point mutations, which, as you should recall, result in a change in the codon sequence. Giving a visual illustration, number 44, translocation, gene amplification, point mutation. In translocation, here we see our proto-oncogene, and these would be our oncogenes down here. In our proto-oncogene, we see translocation, and we see that a section of DNA is relocated within the genome. I guess this doesn't do a great job showing you that, but it shows you here's DNA, this is where the gene is. Now it's been chunked out and moved to a different section. We also see that there's a new promoter here. So this oncogene now at this point, it will now respond to a promoter other than its own. And depending on what this promoter would be used for, it could result in this gene being expressed more than it normally should. So we could have the resulting proteins for this gene being made at this guy's pace, not its own pace. So that's obviously not good. The second would be gene amplification, which is pretty straightforward. A gene is expressed more than it should, and that results in too much protein being produced from this gene as a result. And our third one, point mutations, they could alter the amino acid produced from the codon that it is changed. And I say this because some amino acids have several different codons that code for it. So if this point mutation changes the gene to another codon that codes for the exact same amino acid, it's going to be fine. But if it changes it to something else, well, now it's not so good. Now it's going to be an oncogene. Okay, our bodies are not going to sit back and take all this tumor development business. Hence the function of these tumor suppressor genes. Tumor suppressor genes function as backup. We just have to hope that the mutations in the gene do not happen to the suppressor genes, otherwise they might become cancerous themselves. So how do tumor suppressor genes work? They work in these ways. The tumor suppressor proteins, or the genes, they repair damaged DNA, they control cell adhesion, or cells binding from one to the other, and they inhibit the cell cycle in the signaling pathway. So if these tumor suppressor proteins are not damaged, we can repair some of the things that may have been caused inadvertently previously. But if these are damaged, then we kind of lost our defense system. And our last slide, dealing with 45 and 46 of the reading outline. For the purposes of what we are covering, you will not be filling anything for number 45, that image there. So pay close attention to the discussion on the ROS gene and the P53 gene. I'm going to hold you accountable for those two genes. Also, you should be able to answer question 46, although I will not be directly answering that question for you. One of the well-documented genes is the ROS gene, which is short for rat sarcoma, which is a connective tissue cancer. The protein created by the ROS gene is called the ROS protein, logically, ROS gene, ROS protein. You will have you will know of this as a G protein, G indicating that it is involved with growth. So G proteins are involved with growth of the organism. This protein relays a signal from a growth factor that ultimately causes the cell cycle to get kick-started and create more cells. Hence growth, making more cells. Certain mutations to this gene can lead to rapid, uncontrolled cell division, otherwise known as cancer. In fact, 30% of all human cancers are tied to the mutations in the ROS gene. Another well-documented gene is the suppressor type, and this one is the P53 gene. The 53, if you're wondering, is because of the 53,000 Dalton molecular weight of the protein produced from the P53 gene. When damage has been done to the DNA, the P53 gene becomes active. It produces a protein which prevents the transcription of the damaged DNA until necessary repairs have been made. So it appears to be of the suppressor type. So make sure you take note of the ROS gene and the P53 genes involved in human cancers, and make necessary adjustments to get that information tied to questions 45 and 46. This is it for Chapter 18. We'll be talking about Chapter 19. That will be our last chapter that we discuss for the final exam. Keep in mind, your final exam will be about 50 questions. I'm planning on 50 questions. 20% of those questions are going to be coming from chapters 18 and 19. So that puts it at about 5 questions from chapter 18 and 5 questions from 19.